Okay, welcome. Welcome to Biology Paper 2 revision. Now, honestly, my head is in absolute shambles right now. A lot going on, especially because we have History Paper 3, and then the day after, Biology Paper 2. I'm gonna have to juggle revision a lot here. So the plan is, today we're doing Biology, Monday we're doing History, Tuesday Biology, Wednesday History, and then obviously Thursday I'll try and do some Biology, but obviously, it's the day before the exam. Not going to be much I can do at that point. But what I want to do today is I want to look over a past paper and we revise content. So I'm not going to really look into specific questions. I'm just going to look at the content, see what needs to be revised. Because if we have a look at my big revision document, which is just taking a sec to load. Where is it? Okay, I found it. Took me a sec. Hello. We're not going over witchcraft today, unfortunately. Man, I was gonna yesterday, but the wave of demotivation hit. So I thought, let's switch it up and do biology. Hope you're okay, man. I hope things are all going good with you. So, paper 1 content, we still need to know. Because it could appear on paper 3, and it's still essential to know because it's like the foundation for paper 2. Paper 2, we have energy transfers in between organisms. That's things... Yeah, just switch topics real quick. That's shit like photosynthesis, respiration, anaerobic respiration. So not much. Big processes, but not a lot to know. That's also things like the practical... Oh, thank you, man. Take care. That is also things like the chlorophyll practical, the respiration practical, which are beefy... They're beefy practicals that could appear in paper 2 or 3. So that's a good chunk to know. Then the biggest topic by far is biological processes for homeostasis. Things like the reflex arc, action potentials, Pacinian corpuscle, photoreceptors, myogenic control of a cardiac cycle, synapses, sliding filament hypothesis, second messenger model, and then osmoregulation, which is a beefy chunk. And then obviously we have things like genetics, populations, which is things like speciation, competition, and the nitrogen and phosphorus cycle. And then finally, gene expression, which is a monster of a topic. That's things like transcription control, translation control, reverse transcriptase enzymes, restriction endonucleases, in vivo, in vitro cloning, gel electrophoresis, and DNA probes. A monster of a topic that I am scared out of my mind for. I'm scared to revise for gel electrophoresis and stuff like that. It's a monster of a topic, but anyways, it's fine. It's fine. Okay. Just gonna plug in my laptop and we'll go through. So one thing that I wanna focus on for sure is muscle contraction, sliding filament hypothesis. That's one thing that I want to revise over, because the sliding filament hypothesis is a monster of a topic. Okay. Let's look at question papers and mark schemes. Okay. And it's paper 2. Okay. We will do question paper 2 for 2023. Okay. Oh no, I've already done this paper. Okay. Should we just do 2021? November 2021. Let's hope this is one that we haven't... Okay, this is one we haven't done. First mark. Well, first question. Okay. Describe the process of glycolysis. Okay. Fun question. Glycolysis. So we start off with our 6-carbon compound, glucose. Then thing is, the wording, I always get it wrong. Is it phosphorylates or phosphorylyzes? I'm going to say phosphorylyzes. This is the investment stage where 2 ATP is converted into 2 ADP plus PI. Now, I know what they want you to say here for glycolysis. I know the mark scheme off the top of my head. What they want you to say is that it is a net gain of 2 ATP. It results in the formation of 2 reduced NAD / NADH. That's the other way of writing it. It involves the phosphorylation of glucose, and it is the formation of 2 pyruvates. That's what the mark scheme will want you to say, but we should also just elaborate in detail. So, 6-carbon compound glucose becomes phosphorylated glucose, which then causes it, I guess we'll just do it over here, which then causes it to split into, is it triose phosphate? Then from there, it becomes pyruvate, and that's because we have a redox reaction. So oh shit, how does this work? It's NAD to NADH, which reduces TP to pyruvate, I think. So glucose is phosphorylated by ATP. Phosphorylated glucose breaks into 2 TP. You have to say triose phosphate. You're not allowed to abbreviate unless you later put TP in context. Reduction of NAD. Okay, so yes. Oxidation of 2 TP. Okay, so this becomes oxidized. I forgot about that. And then it's a net gain of 2 ATP. Okay, let's have a look at the mark scheme. Phosphorylation of glucose using ATP. Oxidation of triose phosphate to pyruvate. Net gain of ATP. NAD reduced. Okay, I think that is all marks. Net gain of ATP. Reduction. Phosphorylation of glucose. 2 pyruvates. All I would have needed to add is that it becomes oxidized. Okay. 4 marks, not bad. So remember, glycolysis might seem like a big process. It's not. It's not a big process at all. All you need to know is that glucose becomes phosphorylated. You need to know that the triose phosphate becomes pyruvate via oxidation. You need to know that NAD becomes reduced to NADH, and you need to know that there is a net gain of 2 ATP. That's it. That is all you need to know for glycolysis. Literally just this. And you get 4 marks. That is lovely. And you can draw a diagram too, it says. So you can get marks just for drawing a diagram. And yes, you can say you have to imply a net gain. So 2 are used for produced, that implies a net gain. Cool. Malonate inhibits a reaction in the Krebs cycle. Explain why malonate would decrease the uptake of oxygen in a respiring cell. Oh. Is this going to be that oxygen acts as a terminal acceptor in the electron transport chain? By inhibiting the Krebs cycle, there is no NADH/FADH, so no oxygen can be used in that reaction. Is that what we're leaning towards? Less/no reduced NAD/FAD. Oxygen is the final terminal acceptor. Yes, okay. We got it. Okay, so I want to draw everything. So it's glycolysis. Then it's link. Then it's Krebs. Then it's oxidative. Then it's oxidative phosphorylation. Now, with these processes, glycolysis is going to be the net gain of 2 ATP. It's going to be pyruvate. And it's going to be 2 NADH. Link is also the gain of 2 NADH. Krebs, I think, is the gain of 6. I think it's the gain of 4 NADH. And the gain of 2 FADH. I just want to check on that. Acetate combined, so, oh yeah, obviously, acetyl coenzyme A, but we already knew that. It's decarboxylation in the Krebs cycle. NAD, NAD. I think it must be 6 and 4 then. And then obviously the electron transport chain. Now, where do these occur? These occur in the inner mitochondrial membrane of foldings. So basically the membrane of a matrix. Krebs occurs in the matrix. Link occurs in the matrix. And glycolysis is in the cytoplasm. That's because glycolysis can occur anaerobically. So, let's have a look at the next question. Oh, okay, right. So all of this was useless because it's now asking us a different question entirely. Okay. Great. Just wasted my time doing that. It's good though. It's good to recap that because they are beefy processes. Oxidative phosphorylation, glycolysis, things like the Calvin cycle, LDR, they're all big processes. The more we revise them, the better. And if we get asked an essay question, we can definitively answer it. We can absolutely answer it. If we get a question saying the importance of, I don't know, the importance of energy transfer in organisms, you could talk about the LDR, converting light energy to chemical energy. You could talk about oxidative phosphorylation, etc. Explain how a resting potential is maintained across the axon membrane. Okay, resting potential. So we are going to say that resting potential of minus 70 millivolts. Let's kind of draw this out. Firstly, we have the NaK pump. Na plus K plus pump. This pump pumps out 3 Na plus ions and pumps in 2 K plus ions, leading to a net negative charge inside the axon membrane. Then we have leaky K plus channels, obviously therefore leading to a net negative charge. And we have Na plus channels that are voltage gated, they are closed. Therefore, there can be no re-entry. So let's see what the mark scheme says. Higher concentration of potassium ions inside and higher concentration of sodium ions outside. Potassium ions diffuse out, sodium ions diffuse in. Except sodium ions can't diffuse in due to alternative explanation. Membrane more permeable to potassium ions. Sodium ions actively transported out and sodium ions in. Really? I don't know how many marks we would have got there. Accept 3 Na plus out and 2 in. So yes. But reject if numbers are used incorrectly. Accepts sodium gates / channels are closed. Okay, would have gotten a mark. Accept sodium ions can't diffuse in. Okay. So, one thing I keep forgetting is you have to state the idea of permeability. Okay? So when we're looking at this diagram, the K plus ions are leaving, Na plus can't go back in, Na plus leaving. Therefore, we would say that the membrane is more permeable to K plus ions and not Na plus ions. That is a mark point that I missed, which is odd, but honestly, not bad. I also don't know whether I would have said this in an exam. Diffusion. You always have to mention diffusion. The K plus ions diffuse out. They don't move out, they diffuse out. These are actively transported out. These cannot diffuse in. So that would have been 3 marks, and I think I would have gotten 2. So not bad at all. Explain why the speed of transmission of impulses is faster along a myelinated axon than along a non-myelinated. Okay, I don't know how to explain this fully. All I know is that you have Schwann cells, and inside the little gaps are called nodes of Ranvier. So what you would say is that it allows the electrical impulse to jump between nodes of Ranvier. And I guess you would also say allows depolarization to jump between nodes of Ranvier. Let's have a look at what the mark scheme says. I wouldn't know how else to explain this. Oh, okay, literally 2 marks for stating the obvious. In non-myelinated... Wait, there's no mark? There's no mark for talking about the nodes of Ranvier? Oh, wait. Yes, depolarization at nodes of Ranvier. Would I have gotten a mark for saying allows depolarization to jump between nodes? Surely that's a mark. If I drew this diagram, I might have gotten a mark. You would have had to say depolarization, which I did. Make sure to say depolarization instead of impulses or charge. The 2 marks that I forgot, which is obvious, myelination provides insulation. That's a nice one to remember. Myelination provides insulation. And depolarization occurs across the whole length of the axon in non-myelinated. Because that's the idea, is in non-myelinated, it has to continuously jump the depolarization across the entire length of the axon. In myelinated, it only jumps between the nodes, allowing for a much more faster. So, what you would say is myelination provides insulation. That is a fun word to say. Myelination provides insulation. That is also called saltatory conduction. But, yeah, myelination provides insulation. It allows depolarization to jump between the nodes of Ranvier, and in non-myelinated, the depolarization has to travel the entire length, which is, of course, time-consuming and inefficient. I like that. That was like, I would have gotten a mark, not all of them, but I might have been able to draw a diagram there. A scientist investigated the effect of inhibitors on neurons. She added a respiratory inhibitor to a neuron. The resting potential of a neuron changed from minus 70 MV to 0. Explain why. Okay. This is because no respiration as it's inhibited, which means no ATP production. I'm guessing that would be mark point 1. Mark point 2 is going to be saying that ATP is used in the Na plus K plus pump to maintain a resting potential of 70 millivolts by pumping out free Na plus and pumping in to K plus, because ATP provides energy for active transport of the pump. That's what I think the 3 marks will be getting at. No/less ATP produced. 1 mark. No/less active transport. Electrochemical gradient not maintained. Facilitated diffusion of ions. No net movement of sodium and potassium ions. Okay. I don't know whether I would have said it like that. I would have just said that it maintains a resting potential because of movement of ions. So I guess I would have just had to... Okay, one thing I could have said is it reaches equilibrium. That's why it's at exactly 0 millivolts. I didn't consider that. It reaches an equilibrium. Okay, so again, I would have gotten maybe 2 or 3 marks out of 3 for that. So I'm doing okay at these questions. It's just, I'm doing okay at these questions, just they are winding me up a tad. Some of them have words that you kind of forget. Figure 1 shows an investigation into growth factors of plants. Use your knowledge to explain the growth curvature shown in Figure 1. So this is when the tip is removed and transplanted onto the side like this. And then it curves. The plant tip curves like that. Very crude diagram, I am aware. Use your knowledge of IAA to explain the growth curvature. How would you explain this? Without a directional light source. So, without a directional light source, I'm so confused. Wait, so does that mean there's light everywhere or that there's no light? Would you just say that the IAA is distributed evenly along throughout the tip of the shoot? This is what I would say. IAA is distributed evenly throughout the tip shoot because of the non-directional light. And because it is being added like this, it is going to become top heavy and therefore grow sideways. Tip produces IAA. IAA diffuses into shoot. More elongation of cells on one side than the other. Fuck! That is... Wait, no, that is like an unfair explanation, I feel. That feels like such an unfair explanation, because that is literally just explaining what IAA is. That is such bullshit. What? You can't just pop up some complex-ass diagram and then expect like a grade 1 answer in return. Tip produces IAA. I might have gotten a mark for that. IAA diffuses into shoot. I wouldn't have put that. More elongation of cells on one side than the other. That was a complex, like, confusing diagram. So, the tip produces IAA. 1 mark for that. Jesus Christ. Okay. So if you get asked a confusing question on IAA, just reference the tip producing IAA and then IAA diffusing into the shoot. Because again, this diagram is like odd. I guess what they're trying to convert, what they might be trying to convert is that this is the shoot, this is the tip. It means that only this side is getting, only the left side is getting the elongation, therefore it's going to become heavier. Cells become heavier, which means it's going to fall that way. Okay, so maybe I understand that now. It's that the tip produces IAA, but this side can't diffuse, this side can, so it's like this. I think that that's right. Again, not very nice marking points there, because I would begin to waffle about shit like that, about the unidirectional light, but no, it's literally the tip produces IA diffuses into the shoot, and then IA leads to elongation of cells. Jesus, okay. A bioassay is a method to determine the concentration of a substance by its effect on living tissues. Figure 2 shows the practical procedure used in growth curvature bioassay to determine the concentration of IAA in shoot tips. So they place shoot tips on agar. They remove the agar and cut it into smaller blocks. Each block is placed on one side of a shoot. The shoot is kept in the dark, and a degree of curvature is measured. Describe how you could compare the IAA concentration in shoot tips from two different plant species. In your answer, refer to all variables that should be controlled. Okay, let's try and understand this first. They get an agar, and on it, they put the shoot tips. Obviously, the agar is then going to contain IAA, because as we've just said, the tip produces IAA. Then they cut these into little blocks, each one containing IAA, and they put it on plants only halfway, so only one side is getting the IAA, therefore the plant is going to grow because of cell elongation. So that's the science behind it. Now we need to understand what that actually means. Using the procedure, describe how you could compare IAA concentration in shoot tips. So what it's saying is that the degree of curvature and the concentration in block. And we are comparing the IAA concentration in shoot tips from two different species. So, how would we do this? How you would compare IAA concentration in shoot tips from two different plant species? Obviously, I know what our conclusion is going to be. 5 and 6. It's going to be that the one with the more significant degree of curvature is obviously going to have the higher IAA concentration. All variables that should be controlled. I'm going to guess we would control the time left in the dark. And I guess also just the number of samples to calculate a valid average. And also the time that the tips are left on the agar blocks for, as this will obviously determine the IAA concentration. So that's the idea. The degree of curvature will determine IAA concentration. Let's see what the mark scheme says anyways. So, we need to control the size of the shoot tip, number of shoot tips. Did I say that? I said number of samples. I don't know whether that would count. Size and type of agar, shoots at stem stage, time period they are kept in agar or kept in dark. Okay, would have gotten a mark. Temperature, repeat several times and calculate a mean. Read the degree of curvature to determine IAA concentration. 2 out of 5. That is such a deceptive question. Hold up. I put 5 and 6 here because I expected there to be a whole array of steps before that. But no. All you had to do is refer to variables that should be controlled. I overthought the question and I wrote about the wrong step. That is so unfortunate. So, now we know for next time, things like temperature, the growth stage of each plant, those would be 2 marks. Time left in the dark, yes. The number of shoots, the number of shoot tips, obviously a big mark, which I did begin to highlight, was number of samples. This would then go on to say calculate a valid mean, repeat several times, calculate a mean to then compare. So actually we got the science right. We understood how the science works. What we didn't understand was literally the simplest question. They asked us such a simple question there for 5 marks, and we got 2. Oh no, there's more marks. Oh, I hate questions like this where it just keeps on going on and on and on, because it is so boring. Because it's like, you already know you're doing shit, and then they want you to write more about it. Okay, let's just try and figure this out. So this one is left in the dark. This one, the direction of light is here. A has an impermeable barrier, so nothing can get through, and the direction of light is also coming this way. State 2 conclusions about IAA that you can make from the results shown in Table 1. So, the degree of curvature for 1 and 2 are roughly the same, which implies that the tip produces IAA regardless of light intensity. It produces it in the dark, and it produces a similar amount in the light. Then this one has a way greater concentration than this one, which implies that in order to travel, they must do so via diffusion. IAA is not broken down by light, or IAA is produced in light, or light and dark does not affect IAA production. Yes, that's a mark. Second mark, IAA moves away from light or moves to shaded side. Okay, I said that it needs to move via diffusion. IAA accumulates on shaded side. Idea of movement is required. Okay, I wouldn't have gotten a mark then, but I was on the right path. I just needed to elaborate. All I said was that IAA travels via diffusion. That's kind of obvious. What I needed to say is, this is the shaded side, this is the bright side. It can't travel. It accumulates. I think the idea of a barrier being impermeable kind of stopped me there, because I was like, hold on, how can they move if it's impermeable? Oh shit, I just spoiled a question. It's a fruit fly question, again. In fruit flies, males have the sex chromosome XY. Females have XX. In fruit flies, a gene for the eye colour is carried on the X chromosome. The allele for red eyes is dominant to the allele for white eyes. Okay. So, question 1. is that male fruit flies are more likely to have white eyes. Explain why. Because males only have one X chromosome, therefore they have a greater chance of having a gene where it's like XR, whereas females who have XR are more likely to have a dominant allele to override it. Males have only one allele. Ignore references to X and Y chromosomes. Okay. I guess I'll go fuck myself then. Fuck, okay. So it's this idea of X linkage is males only have one copy of the allele, females have two. Okay, you know what? I'm even going to write that down. I'm even going to write down literally that one sentence in my revision document. Males only have one copy of allele. Females have two copies of allele. Therefore, males more likely to receive whereas females more likely to inherit recessive alleles / females need two alleles. Females could have dominant and recessive. Females must be homozygous recessive. Okay. Sex linkage. So males only have one copy of allele, females have two copies. Males more likely to inherit recessive alleles. Females need two recessive alleles. So they need to be homozygous recessive. A female fruit fly with white eyes was crossed with a male fruit fly with red eyes. Tick one box which correctly describes the phenotypes. Okay. A female with white eyes. So that female has to be lowercase r, lowercase r. Meanwhile, the male is r. Therefore, it's going to always be heterozygous. So it's going to be, they all have red, right? Wait, no. Oh, no, wait, what? Oh, I drew the Punnett square wrong. All females red eyes, all males white. Wait. I would have had to draw it like this. Okay, hold on. X, Y, X, X, R, R, R. So that's going to be XR, XR. That's going to be XR, XR. Oh, okay. So if it's a male, it's going to be white-eyed. If it's a female, it's going to be red-eyed. Okay. Silly me, I drew a simple Punnett square instead of the least obvious Punnett square ever. Fuck off, okay. Come on, I have this stupid... Okay. In fruit flies, the genes for body colour and wing development are not on the sex chromosome. The allele for grey body colour is G. So, grey is G... Black is G. The allele for long wings, L and L. Long, short. A geneticist carried out a cross between fruit flies with grey bodies and long wings. So grey body... and fruit flies with black bodies and short wings. So this. Explain the results on table 2. The results in table 2. So grey body and long wings. We get 2, 2, 2, 3. Black body and short wings. We get 2, 1, 8. So nigh identical. Explain the result. Is it because crossing a heterozygous and a homozygous always results in about 50-50? The 2 genes are linked by autosomal linkage. No crossing over occurs. Only GL uppercase and GL lowercase are produced. What? How were you supposed to infer autosomal linkage? How were you supposed to infer autosomal linkage from that? Am I missing something significant? How were you supposed to infer autosomal linkage? I mean, hey, if I go into an exam and I have no idea what to put for my answer, I guess they've literally just given me a ticket to put autosomal linkage. I don't understand that, though. I don't understand how that would be autosomal linkage. It's this idea that that many are GL, that many are GL. They're not on the sex chromosomes. They crossed them. That's the result of a cross. Explain the results in table 2. No, I don't get this. I don't get this at all. Why would it be autosomal linkage? Can we get a big look into this, please? Alleles are linked. Accept alleles are linked. Oh, it's this idea that there's no things like, there's no G and L / G and L. There's no offspring. There's no gametes of this. There's gametes of this, but only gametes of that. Okay, I'm going to take a screenshot of this. Just going to underline that. I'm going to take a screenshot of this so I remember it. So autosomal linkage is when they are on the same gene, so crossing over can't occur. And that makes sense, because again, it didn't specify anything about them being black-bodied with long wings. There were only 2, when there should have been more combinations. That's a difficult question for 3 marks. I don't know how you would have got autosomal linkage, but okay. The first generation of a population had 50 females. Calculate how many female fruit flies would be produced on the 5th generation. Oh no. Oh fuck, okay. Calculate how many female fruit flies. So firstly, I mean, this is pretty bog standard. XX, XX, XY, XY. It's a 50% chance of it being a female, roughly. And it says that in the question anyways. I'm an idiot, okay. So the first generation has 50. You can assume each female produces 400 offspring each generation. Okay, this might be a bit of a monster of a question to try and answer without a calculator. Okay, so that means 25 of these are going to be female, times by 400. Oh, I'm not good at mental math. 100... Is that 10,000 in the first generation? I really should get a calculator, but I'm lazy. And then I think you would just repeat this, no? So then you would half that number. There's probably an easier way to do this, I am aware. You would just keep doing that until you get, so that's the second generation. No, sorry. That's the second generation. That's the third. That's the fourth. That's the fifth. Let's have a look at what the mark scheme says. It's 1.6 times 10 to the power of 13. Or 8 times 10 to the power of 10. Would that add up? I feel like that makes sense here. Shows 200 to the power of 4. I didn't put that. I don't have a calculator. I would honest to God answer that properly if I had a calculator to answer it with, because I'm not using my laptop. In fact, yeah, I can. Hold on, my laptop has a built-in calculator. Okay, let's try it. So 25 times 400. $10K, yes. Then half of 10K is 5,000 times by 400. That's 2 million offspring. Then 1 million times by 400. Yeah, you can see where this is going. I would need a calculator for that, so I can do it in standard form. How many marks is that? Because that feels like a really nice math question. 3 marks, not bad. Neonatal diabetes is a disease that affects newly born children. The disease is caused by a change in the amino acid sequence. Okay, this could be a nice question. This change prevents insulin binding to its receptor. Explain why this change prevents insulin binding to its receptor. Is that not literally just they are not complementary? Is it literally like tertiary structure of insulin Tertiary structure of insulin is no longer complementary to receptor, therefore cannot bind. Is it literally like that? Change in tertiary structure no longer complementary. Yeah, 2 marks. Lovely. That's a lovely question. Oh, what the hell? Phosphonia... What the hell? Phosphorinositide free kinase P1 free K is an enzyme in several metabolic processes. Figure 5 shows the role in the control of blood glucose. A decrease in the activity of PI3K can cause type 2 diabetes. Wait, is this not the secondary messenger model? Let's draw this out. This is the membrane. So this is the insulin receptor. What we're saying is insulin binds to it. Then we have IRS, which activates PI3K, which then activates AKT. And AKT stimulates the movement of vesicles that contain channel proteins. So this is a vesicle with channel proteins in it, which will obviously fuse with the membrane, allowing glucose to enter. So this is just the secondary messenger model. That's fine. That's a secondary messenger model. A decrease in the activity of PI3K can cause diabetes. Explain why. Okay. So this would literally be saying the role of PI3K is to activate AKT. Without AKT, less or no vesicles containing glucose channel proteins can be stimulated to fuse with the membrane. If that doesn't happen, then glucose remains in the bloodstream as it cannot enter the cell. Therefore, I guess that's kind of it. No AKT activated, yes. So fewer vesicles move to membrane, yes. So fewer glucose channel proteins in membrane, yes. Less glucose diffuses into cell, so high blood glucose. I did not say that. I said that they would remain in the bloodstream. Would have still gotten 2 marks. That was a nice question though. That's literally the secondary messenger model for adrenaline. That's a lovely question. Yeah, it would prevent the activation of this, which would prevent the vesicles fusing with the membrane. That would prevent glucose from entering the cell by diffusing through the channel. That's a lovely question. I would be happy if I got a question on that. So far for this paper, what I'm noticing is I'm not getting 100% for every question. I'm missing 1 or 2 mark points every time, but they're usually pretty obvious. Like even the questions on IAA were pretty obvious. Things that I just missed out on because I overthought the question. This paper is a lot of knowledge, I'm noticing, which is odd because the paper that we're meant to be doing in a couple of days, everyone's predicting is going to be less knowledge. It's going to be more application. Things like evaluating questions, practicals. I feel like this paper's not going to be representative of the one we're doing in a few days, which is why I mean, I'm going to go in a minute anyways. But after this, I'm probably going to try and find a paper that's more about evaluating. That's what I might do, to be honest. because clearly this paper is like 90% knowledge, 10% practicals. I mean, if we have a quick scroll, explain why glucose is found in the urine of a person with diabetes. Hold on. We'll do that in a sec. No, like, most of these are calculation questions. Evaluate the use. Okay, that's a good question. Evaluate the use, and the standard deviation error bars don't overlap. That would be a mark point. Explain how PCR is used to amplify the DNA fragment. Oh, shit. How would you do that? PCR. Oh, fuck, I don't remember that. Oh, PCR is just breaking hydrogen bonds between the two DNA strands, cooling the mixture so that the primers bind. Reaction. Polymerase creates DNA fragments. To form complementary strands. That is a horrible question. Anyways, the question on urine and why it would contain glucose if someone's diabetic is the idea that there's a high concentration of glucose in the blood, which I would have said. Not all glucose can be reabsorbed because the carrier proteins are working at max rate. That's the idea of selective reabsorption of glucose, which occurs in the PCT. That's the idea that the filtrate contains so much glucose that not all of it can be reabsorbed via co-transport because there's so much of it, so all the proteins are saturated. Let me just have a look at one last that mark scheme. DNA polymerase, nucleotides and primers. Heat to break hydrogen bonds. Reduce temperature so primers bind to DNA strands. Increase temperature, DNA polymerase joins nucleotides. Okay. I'm going to copy this so that I replace my answer with it. Okay, so in vitro cloning is literally heat to break hydrogen bonds, separating the strands, reduce so that the primers can bind to the DNA strands, increase temperature so polymerase joins the nucleotides. Okay. Not bad. Anyways, I am going to wrap things up now. Can you revise? So, yes, of course. I'm going to wrap things up now. I like how I just left the word urine on my board. So, clearly, I do have a lot to revise in terms of gene technologies, which I will revise later. I'll go look at some mark schemes. We'll go figure this out properly. I will go figure all of that out so that next time we're here, I can answer some better questions. What I'm going to focus on, my hair, what I'm going to focus on now for the next couple of days is making sure that I'm able to do the evaluation questions, because I know, what is up with my hair, bro? I know that the evaluation questions are going to be lethal. Right, they are usually 5 marks and they are asking you to evaluate something completely unknown. The way you get marks from them is referring to statistical tests, referring to the data, referring to your knowledge, referring to the limits of the testing, things like that. I will make sure that I have that mark scheme locked in my head so that we're okay. Are you doing GCSE? No, we're doing A-level biology, but I'm just wrapping things up now because we've been live for about an hour and I'm tired because I'm lazy. But yeah, what I will do is when it comes to next stream, which I might do one later if I can be bothered, we will focus more on the evaluation and practical questions instead of the knowledge questions. Because while the knowledge questions can trip you up a little, it's more likely than not going to be these practical questions that are the ones tripping me over. I mean, yeah, like, the evaluation questions are literally 5 marks long, and there are dozens of mark points you can get. I mean, yeah. Things like p-values, flaws in the data. Yeah, okay. See you guys in a